This conference will now be recorded.
Hello everyone. Well, I hope everyone is here and we have a lot to cover and we really should begin today's virtual conference. I really appreciate all for attending today's virtual conference. A very good morning, good afternoon or good evening to all the people present here virtually with me today. Let me introduce myself. I am Shirley Prose, Program Manager at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. For the next 5 minutes, please allow me to give a brief description about our organization, Coalesce Research Group, and also the further instructions. Coalesce Research Group LLC is very delighted to welcome you all the speakers supporting associations, students, and delegates present here virtually with us today. With the aim mark the scientific excellence by facilitating conversations, sharing knowledge, Coalesce Research Group follows through the objective of setting standards in delivering excellence in the research among the researchers and industries, promulgating the research through conducting events and online webinars from various fields of science, especially from the stream of medicine, science, engineering, and technology. As a global scientific event organizers, we plan and carry out international conferences, meetings, seminars, symposiums, and workshops providing the best delegate experience, excellent networking opportunities, conversations leading to innovations, learning new opportunities. Just recently, we have also started our own platform for open access peer reviewed scholarly literature where all the content is freely available without any charges to the reader. As being the very new journal, we have coined up some interesting journals and have also formed the editorial board, and we will be very glad if any of our today's attendees, the speakers, students, or delegates could extend your support by checking the journals and will be interested in publishing an inaugural issue at any of the journals according to your area of expertise. We will also send an email with all the details about the journal after the webinar. Do take your time to check the website. Thank you. We also hope your participation will contribute to new research findings and that will lead to new collaborations and research initiatives. Before we start with the sessions, I guess some of you will be new to the GoToMeetings app. Let me just explain you the important tools required for the presentation. On the GoToMeetings app webpage, you can find four icons at the bottom center: mic, camera, screen, and mic button. Click on that to mute and unmute. I request all to stay muted while our speakers are presenting their research work so that it doesn't cause any disturbance to them. Camera button. You can use this button to share or unshare the web camera. If you have the option to share the web camera, you can do it and it is completely your choice. Screen button. You can use this button to share your screen. This is the main part during your presentation. After I call your name from my side, I will be making you the presenter and will give the permission to share your screen. Then only you will be able to share your screen. So once I make you the presenter, you can click on the screen button and you will find a pop-up window to share your screen. And this is your fourth icon where you can use this to leave the session. On the top center, you can use the options to change your display settings. At the top right corner, you will have three icons: people, chat, and settings. In the chat option, you can chat with me or any other participants. For the settings, you can use this button to check for the settings of your speakers and microphones. So as mentioned, I kindly remain muted during the presentation so that the presenters doesn't get disturbed, and for any questions you can ask the questions or you can send your questions in the message box too. Well, let's not take much of your time. Finally, I would like to request you to please give us a brief introduction about yourself before starting with session so that we can know about you virtually. And now let's move into our sessions. The first presentation is by Dr. Ankri Sergey from Ruth and Bruce Report Faculty of Medicine, Israel. Hello, Dr. Hello, Dr. Ankri Sergey. Are you ready with your presentation? Hello, Dr. Ankri Sergey. Are you ready with your presentation? Hello, Dr. Ankri
Sorry, but I
Sergey. Are
don't
you?
hear what you are
Yeah.
saying.
Hello, Dr. Ankri Sergey. Can you hear me? Can you hear me now? Hello. Hello, Dr. Sergey. Can you hear me now? Hello, Dr. Ankri Sergey. Can you hear me? Please unmute yourself. Please unmute yourself.
It's unmute. It should be unmute. Do you hear
Yeah,
me?
yeah, I can hear
Perfect.
you. Yeah, are
Perfect.
you ready with your presentation?
Yeah, no problem. Just try to share it so I don't
Yeah,
know
I'm
because.
making you as a presenter. Please click on the share button to share your screen.
Okay.
Yeah, I made you as a presenter. Please click on the share button to share your screen, please.
Okay. Share. Perfect. And now I'm going to open my presentation. Okay. So do you see the presentation?
No, it's not visible.
It's not visible.
Yeah, yeah, it's not visible.
Just a moment.
Hel
Do you see it?
yeah, it's perfect. Please make it a full screen and start your presentation.
And now, do you see it?
Yeah,
Because
please
I
make it a
see.
full screen. Please make it a full
Okay.
screen and
This is
start your
what I have
presentation.
done. Just a moment, because this is what I have done. Okay. So full screen. Okay. So do you see now the presentation full screen?
No, no, please make it a full screen.
Yeah, maybe you can advise because usually I'm just pushing here and I can make it full screen. So view, maybe view, fit to Windows, maybe, no. Uh, how to do that?
Um, can you on the top you can find slideshow option. Can you please click on it?
Slideshow, yeah. From the
Yeah,
beginning, yeah.
y-
This is
yeah.
what it gives me.
Please
So
double click on it.
No, it does not work. I mean, this is what my
Please,
computer is giving.
okay, please go to display settings on the top.
Okay. Swap, maybe?
Yeah, please click on
Oh,
the slideshow. Yeah,
I
it's
think it should be
perfect.
okay.
Please continue. Yeah, that's perfect. Please continue with your presentation.
Okay, thank you very much. So first, I would like to thank the organizer for agreeing to shift my talk to this virtual talk and allowing me to present my work at this fifth European Congress on Infectious Disease. So my lab is located in Israel, in Haifa, at the Technion University, and I'm a parasitologist working on the parasite Entamoeba histolytica. And today I'm going to tell you three stories dealing with the interaction of the parasite Entamoeba histolytica with the gut microbiota. So let's begin by some facts about amoebiasis. This is a disease caused by the parasite Entamoeba histolytica. This is a unicellular parasite that infects humans. The prevalence of the Entamoeba infection is high. It has 50% in areas of Central and South America, Africa, and Asia. And it is considered to be the second leading cause of death due to parasitic disease. In most cases, the infection is asymptomatic. However, in around 10% of the patient will develop bloody diarrhea, with here you can see the ulceration of the large intestine. And in around 0.5 to 0.1% of the patient, the parasite escape the large intestine to reach the liver, where it will form these liver abscesses. So you get infected by the cyst, the infective form of the parasite present into fecal material. So it's a fecal contamination. The cyst released the trophozoite in the small intestine. The trophozoite colonized then the large intestine. In most of the cases, the story stops here. The trophozoite feed on the gut microbiota, form new cyst, release into the feces, and that's all. As I mentioned, in some cases, the parasite is able to destroy the mucus layer, destroy the intestinal epithelial cells, reach the lamina propria, where it will trigger a very strong inflammatory response, characterized by the recruitment of macrophage and neutrophiles. And all of these cells are producing reactive oxygen species and nitric oxide, which are very toxic for the parasite. And in my lab, we are really interested to understand how the parasite sustain these stresses. But today, I'm going to focus on the interaction, as mentioned, of the parasite with the gut microbiota. Before, I would like to indicate that there is a very efficient drug against amoeba called metronidazole drugs or flagyl. This is a drug activated by thyroidoxine reductase inside the parasite to form a nitroradical anion, which is very, very reactive, causing oxidative stress inside the cells, and it can even cross-react with DNA of the parasite, breaking the DNA. Also, the metronidazole can be interacting with very important protein, which are involved for redox detoxification inside the parasite, causing adduct and inhibition of this enzyme, leading to the death of the parasite. So this is a very efficient drug against the parasite, but in the field, mostly in India, it has been found that patients were resistant somewhat to the drugs, and in vitro, we showed clearly that the parasite can adapt to some toxic level of metronidazole. Indeed, this drug, flagyl, has some side effects. It causes headache, diarrhea, loss of appetite, so many side effect that I have myself experimented when I got infected by this kind of parasite during travel in India. So this slide summarizes what is known about the interaction between the host microbiota and the parasite Entamoeba histolytica. For example, in the 80s, it has been found that the short-term interaction between histolytica and the bacteria E. coli enteropathogenic O55, it will lead to a boost in the virulence of the parasite. We know also from metagenomic analysis that in patients that develop liver abscesses, there is an increase in Klebsiella inside the population of the bacteria present into the gut. The parasite feed also on bacteria, causing dysbiosis. One highlight of this dysbiosis is the increase of Bifidobacterium bacteria and decrease of the Bacteroides. So this slide shows you that there is a certain knowledge on what the bacteria are doing to the parasite and vice versa, and how these bacteria are affecting the outcome of the disease. So in my lab, we are investigating this cross talk between the bacteria and the gut and the parasite, and I'm going to tell you three stories. Two will be focused on compounds produced by the gut microbiota and how they affect resistance to stress inside the parasite. And the last story that just been published today in NPG Biofilms and Microbiome will focus in the interaction between the parasite Entamoeba histolytica and biofilm. Okay, so everything began here when we incubated the amoeba with a bacteria E. coli K120055. And what we found is that the parasite is very sensitive to oxidative stress caused by hydrogen peroxide. But when you have bacteria in the culture, the parasite is protected. So we show that even if you separate with a filter the bacteria from the parasite, you can see this protective effect taking place, meaning that there is small molecules going into the culture produced by the bacteria, and they are protecting the parasite against oxidative stress. So what we did, to make a long story short, we screened a library of E. coli mutants, and what we found is that one of these mutants, one that was knocked out for the malate dehydrogenase enzyme, was unable to protect the parasite against oxidative stress. However, if you complement the bacteria with a wild-type MDH gene, then the bacteria is again able to confer to the amoeba this resistance to oxidative stress. So what is malate dehydrogenase? This is an enzyme that takes malate and it converts it to oxaloacetate. And we are very excited to see in the literature that this oxaloacetate can detoxify hydrogen peroxide to form CO2 and H2O. So based on this evidence, we built the following experiment. We exposed the amoeba to hydrogen peroxide with and without oxaloacetate, and you can see that nicely oxaloacetate is protecting the parasite against oxidative stress. You cannot recapitulate this effect if you use the malate, the substrate of the malate dehydrogenase, or the enzyme alone. But if you combine malate and malate dehydrogenase, then you produce oxaloacetate and you can recapitulate this nice protective effect. Finally, we use the mice model that we are using to develop amoebiasis. In that model, the parasite is directly injected into the cecum of the mice, and we inject it with and without oxaloacetate. And you can see clearly that when the parasite is injected with oxaloacetate, you have a better invisibility and infection of the mice model. So the oxaloacetate is clearly boosting the virulence even in the mice model. So to summarize a small story here, what we think is that when the parasite meet a strong oxidant like hydrogen peroxide into the gut as a response to these neutrophiles and macrophage, it used the bacteria in its environment that are producing oxaloacetate to shield itself against this hydrogen peroxide. That can be done directly or by MDH secreted by the bacteria, and they use malate present in abundance inside the large intestine to produce this oxaloacetate that can detoxify hydrogen peroxide. So I'm done with the first story, and now I'm going to talk about a second story that also involves a small molecule cuin, which is very interesting molecule produced by the gut microbiota. And it deals with epitranscriptomics regulation. You know that the Nobel Prize has been given this year on a messenger RNA modification. And this modification on the RNA world are now becoming with such importance, are they really crucial to regulate many functions, including stress response. So in my lab, we are focusing on tRNA modification, and with the help of Professor Didon at the MIT, we look at all the modification happening into tRNA, And there is tens of this modification, but in the talk, I will focus only on the queuing modification because, as I mentioned, this queuing is produced exclusively by the gut microbiota. In human, we have our tRNA modified, but we get this queuing from our gut microbiota or from our food. So what the queuing is doing, if you have a tRNA, there is only some species that are modified. I don't want to develop too much. This tRNA will be modified at the G34 with this enzyme, the TGT enzyme, that uses the queuing as a substrate, and then it will modify it to a Q34. Now, this tRNA, when it is Q34 modified, it's become a better substrate for a DNMT2 enzyme that is going to methylate at the C38 location in the tRNA. And now, this tRNA becomes much more stronger against cleavage from nuclease, and it will be used preferentially for what? For translation of protein involved in stress resistance. So this is how it works in human, but in amoeba we had absolutely no idea about what this queuing is doing to the parasite. The first experiment was to take the parasite to incubate it with synthetic queuing, and by doing HPLC mass spectrometry, you can see that when the parasite have queuing in the growth culture, immediately you have accumulation of queuing into the tRNA. However, this does not happen if you silence the TGT genes, which is involved in taking the queuing and incorporating it into the tRNA. So we saw that the queuing provides a mild but significant protection of the parasite against oxidative stress. And of course, this does not happen in a mutant that have TGT silence. Then we move to the mice model again, and we inject the amoeba inside the cecum with and without queuing. And to our surprise, we found that in presence of queuing, the parasite have a lower ability to colonize the mice. Let's try to make some order here because I feel that you may be confused. I told you that the queuing provide a mild response to oxidative stress, so you may think that the amoeba will be better, virulent, more virulent inside the mice model. But in fact, you have to come back to the first slide I showed you and what I told you about the disease. In most of the cases, the infection is asymptomatic. And what we are thinking is that when the parasite is in presence of a gut microbiota providing queuing, then this gut microbiota is going to give to the parasite a slight advantage in terms of resistance to oxidative stress, but it will cause price a decrease in the virulence of the parasite, so the parasite can stay longer inside the host, enjoying its life, feeding on bacteria, but not causing disease. However, in cases where a strong inflammatory response is triggered, still we don't know why, maybe because of some change in the microbiota composition, then the parasite needs some mechanism to protect itself against oxidative stress. And here is using oxaloacetate, as I mentioned in the first story, from the bacteria that can provide this molecule that can detoxify hydrogen peroxide. So I hope that I cleared the point here. And to continue on the queuing story, we performed transcriptomics analysis and we checked the genes that were upregulated or downregulated when the parasite is exposed to queuing. And what we found is that many genes involved in stress response, in redox regulation, like oxidoreductase, like DNA repairing protein, were upregulated. So the queuing really prepares the amoeba to resist oxidative stress. On the other hand, many genes involved in virulence, like the cysteine protease, that are really crucial for the parasite to destroy human tissue, had their expression downregulated. Okay, so what I told you here is that the synthetic queuing can do many things inside the parasite, but in real, inside the gut, the amoeba is not getting synthetic queuing. It's getting the queuing from the bacteria. So we wanted to know if the parasite have a salvage pathway for the queuing from E. coli that we use as a model. So in this experiment, we incubated the amoeba with E. coli bacteria for 1 week. extract the RNA, and we perform an APB gel. So what is an APB gel? Here you can see on this APB gel a species of tRNA that is not modified by queuing. And if it is, you have unnatural bands. So consider here non-modified tRNA and here modified tRNA. So in the wild-type amoeba growing without queuing, no tRNA Q modified. Synthetic queuing added to the culture, this is a positive control, tRNA is modified. And of course, very excitingly, when you add E. coli K12 to the culture, you can see that the tRNA now becomes modified. But this is not happening when you use an E. coli mutant that cannot produce queuing. So indeed, the amoeba can salvage queuing from the bacteria. So to make a long story short, we were able to find a glycosylase called DEF2419. This enzyme that we characterize deeply can take the querosene, convert it to the queuine, and then this queuine is used by the TGT to be incorporated into the tRNA of the parasite. We silence the expression of DEF2419, and you just have to look at this APB gel here. The amoeba silence for the DEF2419 cannot salvage the queuine from the bacteria. So now, if we want to summarize the second part, we know that the amoeba get its queuine directly from the bacteria. It can phagocytize the bacteria. It can salvage with the DEF2419 the querosene to transform it into queuine. And then the queuine goes inside the tRNA via the TGT enzyme. The qtRNA modified has an effect on translation of stress response protein. And we know also that the qtRNA modified situation is more fitting to an asymptomatic version of the parasite when it is inside the host with decrease in virulence and a mild resistance to oxidative stress. Okay. I hope I'm on time because I'm not following my watch, but now I'm going to talk about the last part of the project. Here will be the interaction of the parasite with biofilm form of bacteria. Until now, all the study that has been done in the field has focused on bacteria, isolated bacteria, which are called planktonic form. So what happens when an amoeba meets a biofilm? So for this study, we studied two bacteria that are able to form biofilms, B. subtilis and E. coli. So we are going to focus mostly on a single species biofilm, which are quite very convenient, as I will explain in one moment. So what are biofilm? Biofilm are bacteria bound together and bound to a support. So they form aggregate and they are bound to a support. That was the old definition. Recently, a paper in Nature, I think, showed that even small aggregates that are floating are considered biofilm. So the definition changed a little. That will be important for the continuation of the story. So these biofilms are really a nightmare. I don't have to anticipate to this audience what the biofilms are doing. They are causing resistance to antibiotic. In clinic, they are going emergence of disease. In industry, they are clogging tubes, pipes. These are just nightmare. So in the large intestine, there is clear evidence of presence of biofilm. Even by coloscopy, you can see them in people infected with inflammatory bowel disease. So here are summarized all the bacteria found in this biofilm. However, there is a strong controversy about the presence of biofilm in the healthy gut. Until this paper that showed that small aggregates can be present in healthy gut, and you can detect the biofilm at the condition that you use mild condition for the fixation of the tissue. In fact, many people missed this biofilm because the condition to fix the tissue were too harsh. So here you can see the intestinal epithelial cells. It's a green orange staining, and on the top you can see a biofilm of bacteria. So no doubt that you have this biofilm present even on healthy guts. So as I mentioned, we use B. subtilis biofilm as a model. Why? Because first, there are probiotics present into the gut of human. They are used for fermentation of food in Asiatic countries. And they are a very simple model where the TAS-A protein has been perfectly characterized. This is the glue that allows the bacteria to stick together. You can see here by electron microscopy, or the TASA join the bacteria together. So this protein is really important for biofilm formation. So we know what is the main protein here involved in the structuration of the biofilm. So in this experiment, we used amoebic media where we grow a biofilm of B. subtilis. Here you can see in microscopy that this B. subtilis beautiful biofilm is expressing GFP. I will tell you in one moment what it's important. And then we pour a culture of antibodies tolytica on this biofilm, and we follow a disappearance of the GFP signal that will translate into a degradation of the biofilm. Using this approach, we found that the biofilm is destroyed under a dose and time dependent, meaning when you put more amoeba, you have more biofilm destroyed. When you incubate longer time, more biofilm is destroyed. So that was quite very nice to see this observation and to visualize this destruction of the biofilm. You can see here in electron microscopy in gray, a nice biofilm. In black, the amoeba. And here you can see a hole. The amoeba destroyed the biofilm. In immunofluorescence microscopy, you can see in green the amoeba on the biofilm. In black here, some zone of destruction. And if you look even in 3D, the amoeba, penetrate inside the biofilm. And if you can visualize the red spot here, these are bacteria that has been phagocytized by the amoeba and after being detached from the biofilm. So we perform a transcriptomic analysis to see how the parasite behave in terms of transcriptomes when it is incubated with biofilm versus planktonic form of the bacteria. What you can see here is that the transcriptomic response of Histolytica with B. subtilis biofilm is very different from the transcriptomes of the amoeba in incubation with planktonic form of the bacteria or without bacteria. So that was very exciting because that was the first time that such behavior been observed on an amoeba. And when we analyzed the data, we found that many cysteine proteinase genes that I remind you are very important for the degradation of human tissue were upregulated. However, genes involved in redox regulation were downregulated. So we use an inhibitor of cysteine proteinase called E64D, and we also silence the expression of CP5, which is a major surface cysteine proteinase in antibodies tolytica. And in both cases, you can see that the degradation of the B. subtilis biofilm is inhibited. So these cysteine proteinase that were upregulated are very important for the degradation of the biofilm. I mentioned that TASA, the proteinous glue inside the biofilm of B. subtilis, is really important for the maintenance of the biofilm. Here you have just the visualization of this important. This is a mutant of B. subtilis that does not produce TASA, no biofilm, compared to the wild type. Okay? So we had the chance to have a collaboration with Pedro Rodriguez, and we got recombinant TASA from B. subtilis, pure TASA. When you incubate TASA with a lysate of amoeba, you see that the band of TASA completely disappeared because this TASA is degraded by the amoebic cysteine proteinase. If you do the same experiment in presence of the cysteine proteinase inhibitor E64D, the TASA is not degraded. So this experiment showed that TASA is cleaved by the amoebic cysteine proteinase. Then we focused on the second part of the transcriptomics, the downregulation of genes involved in stress response. Why this is happening? In fact, into a biofilm, you have a gradient of oxygen. This is very well known. And we think that the parasite, when it penetrates inside the biofilm, it becomes protected against oxidative stress. Just maybe forget to mention that amoeba are anaerobic pathogens. So to visualize if this is really the case, we took amoeba and we incubate them with a probe that is sensitive to reactive oxygen species. So when the amoeba turns green, it means it has a lot of reactive oxygen species. What you can see here are amoeba inside the biofilm, and you can really visualize that when the amoeba penetrate inside the biofilm, okay, inside they are less fluorescent, and we quantify that, and you can see that it's significant. When the parasite enter into the biofilm, they are less exposed to reactive oxygen species. And finally, the last experiment here, we ask if a biofilm can inhibit the progression of amoeba toward mammalian cells, because the amoeba know perfectly well to destroy these mammalian cells. So do we see here an inhibition of the destruction of amoeba, of the mammalian cells by the amoeba when there is a biofilm? And indeed, this is the case. If you get trophozoite alone, they are perfectly able to destroy this layer of HeLa cells. If you incubate the amoeba with planktonic form of B. subtilis, they can still destroy these HeLa cells. But if you add on the top a biofilm, you can see that the amoeba are impaired in their progression and they cannot destroy these mammalian cells. Of course, this is something done in vitro, and we are now interested to do the same experiment in vivo. And finally, I just want to emphasize that this conclusion on the ability of the amoeba to destroy B. subtilis biofilm are also valid with E. coli, Enterococcus fecalis, Salmonella. We tried many bacteria, and all the bacteria we tested had their biofilm destroyed by the parasite in a cysteine proteinase dependent manner, which is very interesting because here we have a very potent enzyme able to destroy many biofilms. So in summary, what I showed you today is that the amoeba knows to recognize a biofilm. It can destroy it using cysteine proteinase. In the case of B. subtilis biofilm, we were now perfectly able to distinguish the target. This is a TAS-A protein. As I mentioned 2 minutes ago, many biofilms from different bacteria can be destroyed by the amoeba, and the biofilm protect the amoeba against oxidative stress. It can be due to a gradient of oxygen, but maybe the biofilm is producing antioxidant molecules that we are now in the process of investigation. And finally, the biofilm protects the human cells against destruction by the parasite by forming a shield that impairs the progression of the amoeba. So briefly, what next? All what I told you today is focused on single species biofilm, and now we have developed a humanized mice model that produce inside the gut a gut microbiota from human, and we were able to produce biofilm from the feces of these mice or from cecal material. So we have now at our disposition biofilm from humanized mice that are producing biofilm from multi-species bacteria. And we have preliminary evidence that the amoeba also know perfectly how to destroy this multi-species biofilm. It's very exciting. And finally, I just want to emphasize that we are now a parasite that is really a gold mine for anti-biofilm molecules. We focus on the cysteine proteinase, but in the future, we expect to find other molecules that are able to destroy this biofilm with in mind not only medical application, but also probably industrial application, clinical application, and so on to help destroy this biofilm. So I would like to acknowledge at the end the lab members and specifically a very talented PhD student, Eva Zandinetas, that did all this work. We had fantastic collaborators that help us in this project, specifically Dr. Ilana Kolotin-Gal, that really help us progressing with the biology of the biofilm of B. subtilis and many other studies that I didn't mention in this talk because of the time. And I would like to thank the funding agencies that support this work. And thank you very much for your attention.
Thank you, Dr. Ankri Sergey. That's a wonderful presentation. Does anyone have any query?
No question. Everything was so clear. Happy.
Okay. No, I hope there
Oh,
are no queries.
thank you very much.
Thank you once again, Dr. Ankri Sergey. That's a
My
wonderful
pleasure.
presentation.
My pleasure.
Yeah. So let's move on to the next presentation. The next presentation is by Dr. Vladimir Zazak from Cancer Research Institute, Slovakia. Hello, Dr. Vladimir. Are you ready with your presentation?
Hello,
Hello,
I'm
doc.
here.
Hello, doc.
Okay. Uh, Excuse me, I hear very bad. Okay. Hello,
Hello.
do you
Hello,
hear
Dr.
me?
Vladimir. Yeah, you're audible.
Okay, and I
Yeah, I'm
may
making you. Yes, I'm making you as
start.
a presenter. Please click on the share button to share your screen, please.
Okay. Hello, hello.
Hello, doc.
May I start?
Yeah. Please click on the share button to share your screen, please.
Uh,
I made you as a
do,
presenter.
do you see my slides? Hello? Yes. May
No,
I
doctor,
may I
it's not visible.
may I start my presentation?
No, it's not. Your screen is not visible.
Sorry, I don't understand. Speak slowly.
Your screen is not visible.
So I start my presentation. Okay.
We cannot see your screen. Your screen is not visible.
You cannot see my screen.
Yes.
You see my screen or not?
No, we cannot. We are unable to see your screen.
Do you see my screen or not?
No, we are unable to see your screen.
Hello,
Hello,
may I start
hello,
presentation?
doctor. I- We are unable to see your screen. Please click on the
Do you
share
see my
button
screen?
to share your screen.
Do you see my screen or not?
No, Dr. Zazak, we are unable to
Hello,
see your screen. Hello, Dr. Zazak. Can you hear me?
please. I Do you see my presentation?
No,
Hello,
your no presentation is not visible. Please check. Please click on the share button to share your screen.
so I will start my presentation. Okay.
Hello, Dr. Zazak. Your presentation is not visible. Please check your screen. Please click on the share button to share your screen.
Boże moje, caramba. I have problem. There is some problem. I am sorry. There is something wrong.
He- Hello everyone, there is some technical problem with Dr. Zizak's end, so let us wait for him for 5 more minutes.
Hello, hello. I don't know if I.
Hello, hello Dr. Zizak. Yeah, you're audible.
This conference will now be recorded.
Hello everyone, I think there is a technical problem with Dr. Zizak, so let us move on to the next presentation. So the next presentation is by Dr. Butel Orhan Kelik from Turkey. So Dr. Butel, are you ready with your presentation?
Yes, I'm right.
Yeah, so
I'm
I'm
making.
making you as a I'm making you as a presenter. Please click on the share button to share your screen, please.
Okay, I can. I try to share. Do you want to see my presentation?
Yeah, yes, your screen is visible. It's perfect, and please continue with the presentation.
Okay, can I begin?
Yes, please continue with your presentation.
Okay. Good afternoon, everybody. I want to thank you, Congress Commit, this valuable results. And I will present now our study titled Varicella Seroprevalence in Pediatric Population, results from our single center study. And I want to give some information about varicella. Varicella, we know, is a highly contagious illness with potentially severe complications, especially in young children. The administration of varicella vaccine, this is an effective preventive measure to significantly reduce incidence of varicella. We know before the widespread availability of vaccine, Europe reported approximately 5.5 billion varicella cases, with 18% of unvaccinated children and adolescents showing positive serum anti-VCV IgG. Similarly, in the United States, approximately 4 billion cases were reported each year before the vaccine became widely accessible. However, since the introduction of single-dose vaccination for children aged 12 to 18 months in 1996, the incidence of varicella decreased about 19 by 2005. And what about the vaccination? The World Health Organization and the Centers for Disease Control and Prevention has recommended the inclusion of varicella vaccine in routine childhood immunization programs. This includes 2 doses of varicella vaccine. The first one is typically administered between the 12-15 months of age, and the second dose given the children aged to 4 to 6 years. And what about varicella vaccination strategy in our country, Turkey? The varicella vaccine in the national vaccination calendar since 2013. According to the vaccination schedule, you can see our vaccination schedule. Children in Turkey receive the varicella vaccine at the age of 1 year, only single dose. Private vaccination are available at the age of 4 if parents wish to give their children a second dose of chickenpox. Despite the success of varicella vaccination programs in reducing the incidence of disease, cases of varicella infection continue to occur both domestically and internationally, as reported in several studies. And I will say something. While a single dose of vaccine has been shown to provide approximately 97% protection against severe and moderate chickenpox infection, its efficacy against any degree of severity is lower, ranging from 80 than 85%. Therefore, it is possible for children who receive a single dose of vaccine to still contract a chickenpox infection. In contrast, administering 2 doses of chickenpox vaccine has been reported to significantly reduce the incidence of chickenpox cases. What about our study aim? We aim to assess the prevalence of varicella antibody seropositivity in healthy children for then 12 years who had a single dose of varicella vaccine at the age of 1 year. Our study, retrospective study, we included the children above 4 years old. Their varicella antibody had been checked within the 2 years, and their parents, whose parents sought to verify varicella serology. The other characteristic of these children regularly visited in our clinic well child checkups. This study received approval from Başkent University Faculty of Medicine, and then we conducted this. We analyzed the data according to the age group. We classified two groups, 4 then 6 H1 group, 7 then 12 H2 group. This is school age. Then we analyze IBM SPSS and we done multiple logistic regression analysis because due to investigate the factors influencing varicella seropositivity rate. And what about the varicella zoster IgG antibodies? The enzyme-like immune sorbent assay test kit was used to determine the presence antibody in the samples. Samples with delta value below the threshold was classified as negative and above the threshold were classified as positive. Let's look at the study design. We reviewed total 90 children, but some are there excluded due to 5 excluded due to missing vaccination information and 4 excluded due to pre-existing chronic illnesses. Total 81 children included. All of them, only you can see here 58%, 47 children varicella seropositive, while 42% children varicella seronegative. All seronegative ones were vaccinated without cost. And let's look our results. The study includes, I said before, 81 healthy children with an age of nearly 6. We can look gender distribution here. Female 43:1 female, 38:1 male. And if we looked about what about the age distribution, you can see most of them 75% were aged between 4 and 6 years, and nearly 24% were aged between 7 to 12 years old. The distribution of gender was similar, both age groups. It is important for us. And what about the varicella seronegative and positives comparing? If we compare this, we show there is no differences mean age. You see these are mean age of our children. Seronegative nearly 7, seropositive 5, but there is no significant. But if we compare them according to the age group, we found a significant differences between them. We can see in the 4 and 6 group was found, you can see here 67% here, and while the rate 7 to 12 age was group only 30. And no significant differences was found in terms of gender, you can see here. And this table shows us univariate and multivariate regression analysis, which we aim to identify variables to predict varicella seropositivity rates. And you can see here gender, no differences between them, no significant influence, but we show the results show the age group, especially for the 6 years age group, was a significant predictive of varicella seropositivity rates. And multivariate, as a result of multivariate logistic regression, remained positive because gender is not wasn't a significant predictive factor. Varicella, commonly known as chickenpox, is a viral infection that can have severe and life-threatening complications in children. It is known that varicella vaccination is one of the most effective ways to protect against this disease. The current study reported healthy children, varicella seropositivity rate 58% among healthy children in our center. And several studies have been conducted to assess varicella seroprevalence and the impact vaccination policies in different countries. Let's look some other research. Bolas et al. Conducted a systemic review of 43 studies in European countries. They found considerable variation in varicella seroprevalence among children, emphasizing the need to tolerate vaccination policy, they said. Such et al. This research conducted in our country, and they contrast to our study, they said high seroprevalence of varicella. Who et al. Investigated varicella vaccination in China's children. They think like us. They found low to dose vaccination rates among these of history varicella infection, and they recommending increasing coverage and two-dose regime in the national immunization program of China. Another important finding our study a significant differences in varicella IgG positivity rate between the 4 then 7 age group and the 7 then 12 age group, with a higher rate observed in younger age. We found similar to our study, Gabittu et al. Conducted in Italy, they found high overall seroprevalence varicella with increasing seropositivity in younger age after the implementation of mandatory varicella vaccination for newborns, they said. What about our study? Zhang et al. A study, they found a significant increase in positive antibody, which increasing number of vaccine doses administered and higher protection rate with two-dose vaccination compared to one-dose vaccination. Luan found a higher seroprevalence anti-VCV IgG antibodies in children who had received varicella vaccination, indicating a positive impact of vaccination on the development of immunity against varicella. However, vaccination rates difference between genders, they said, highlighting the need for targeted vaccination strategies. All of these studies, we believed these studies highlight the influence of factors like age, gender, vaccination status, and geographical location on varicella seropositivity rates. Tolerated vaccination policies based on local epidemiological data are necessary to address the variation in varicella seroprevalence. And we believe that further research is needed to understand the factors contributing to heterogeneity and identify optimal vaccination strategy for different populations. Although this valuable contribution, our study some had limitation, such as small sample size. This is a retrospective study, and the lack of examination antibody levels in healthy children in adolescents. Only single center experience doesn't show us maybe community status. In conclusion, while we acknowledge the limitation of our study, we believe that our finding provides important insight into the cerebroprevalence in healthy children in our country and can serve as a starting point for further research in this area. We hope that our study will encourage continued investigation into the epidemiologic varicella and inform the development of effective vaccination programs to reduce the burden of this infection in our community and beyond. Thank for your attention. Thank you for listening to me.
Thank you, Dr. Butel Oren. That's a wonderful presentation. Does anyone have any query?
Okay, I think.
No queries. Let's move forward with the further presentations. Thank you once again, Dr. Beutel Oren, for your wonderful presentation. Thank you.
My pleasure. Thank you very much.
And the next presentation is by Dr. Vladimir Zizak from Cancer Research Institute, Slovakia. Hello, Dr. Vladimir. Are you ready with your presentation?
Hello.
Hello.
Uh,
Yes. So you're audible, and I am making you as a presenter.
Please be so kind and apply my presentation I send you by mail. Okay?
Okay. Okay. Yes, we will play from our end, and you can explain it. Okay.
Okay. Okay.
Hello, Dr.
Yeah.
Zizak. Your presentation is visible now. Please continue with your presentation and instruct us with the next slide so that we'll move the next slide from our end.
Uh, but may you change me slide? I cannot do it. Next slide, please. Okay. May I start? Hello.
Yes,
May I
yes,
go?
Dr. Zizak. Yes, Dr.
Hey,
Zizak.
yes.
Please continue with your presentation.
Okay. Okay. Every virus is a paradigm that can exist on its own. It fully depends on its carrier. This is a basic condition of its existence. The host of the parasite is a living cell, but it generally claims that the virus can exist for 2 to 5 seconds, during which it is transmitted on other species. What living cells carry viruses? Based on work with mobile leukemia virus in a stable, we concluded that the bacterial cells can be the host of the virus. We tested this assumption and confirmed the result. This idea was then tested on HIV model in the laboratory of Professor Flossie von Stahl, UCSD. Next slide, please. Next slide, please. Hello. Okay. Even with this virus, we have been able to prove that its host may be bacteria. Evidence was confirmed at the DNA level and on the protein level as well. In nasopharyngeal swab of HIV-positive children from Cambodia and Kenya, HIV was found in bacteria, but also was found in yeast Candida albicans. Based on this result, it was concluded that many, if not all, viruses can be transmitted by bacteria or by yeast. If all, the coronavirus. A virus like a parasite is not a full-featured biological form, and thus, sorry, okay, and thus hard to fight. Its main weakness is that it's a feel hosted by carriers. Next slide, please. Here you can see PCR product 1526 base pair synthesized on template of bacterial DNA of AIDS patient. Next slide, please. Here is a Western blotting of protein isolated from bacteria in yeast of respiratory tract of Cambodian and Kenyan HIV-positive children. Next slide, please. Here is internalization of normal human lymphocytes by bacteria of AIDS patient. You can see that complete lysis of human lymphocyte mostly. Next slide, please. Analysis of possible participation of bacteria bearing HIV in immunodeficiency, reduction of the amount quantity in intestinal tract of HIV AIDS patient was performed by parallel application of probiotic bacteria E. coli strain NISL 1997. For this study, 18 HIV-infected RT-naive patients were selected. The probiotic were applied per diem in a period of 3 months. The presence of probiotic bacteria in patient intestinal tract was checked by PCR. After 3 months of probiotic application, the viral load decreased or remained on the detection level at 57.5% of tested patients and completely trimmed down about 67%. The viral load of the control group of 8 asymptomatic patients increased by 77% over the corresponding time. Next slide, please. Hello? There is again a problem. Yeah, bacteria is a complete biological form and they can be eliminated by destroying the vector carrying the virus. The virus ceases to exist. Thus, many viral infections can be stopped. It is necessary to find out which carrier used the virus. It's likely that HIV is transmitted to human and travels far to the recipient cells of the hematopoietic system. Upon contact of the viral tentacles with the CD4 receptor, the virus is penetrate the recipient cells and process of tissue destruction begins, thus overcoming the infection and eliminating the virus in the recipient cells by conventional drug-based treatment approach such as heart and activation of the immune system. The infection is suppressed and patient can be pronounced cured. Next slide, please. The virus leaves the hematopoietic system after this therapy, but virus-containing carrier can establish themselves in the intestinal tract and multiply under optimal conditions. Thus, individuals after infection can be infected again, and after penetration of the virus from intestinal tract into the body and also infect other individuals by secreting HIV from mouth, nose, but also from the rectum in a stool. Surprisingly, no attention is currently being paid to a possible fecal infection. This important approach has been proven many times in history and has helped to overcome epidemics. It's surprising why in the current epidemic this possibility of transmitting infection is absolutely marginal and has not been considered. Next slide, please. We have become better at successfully preventing the droplet aspect of the infection. However, we are not preventing the fecal infection yet. It is necessary to introduce early and effective disposal of excrement and disinfection of the toilet by UV lamp ventilation, and finally washing or disinfection of the hand. These measures should be taken where there are shared toilet. However, if the virus enters the intestinal tract, vaccines are not effective here, which is generally the case for all viral infections. For this reason, a reliable and effective HIV vaccine has not been developed yet. This is because the main reservoir of the virus is in intestinal tract and not in the cells of hematopoietic system. According to our previous result in HIV system, the virus remains in the body, but those of established in intestinal tract may exist here in a carrier for years and infect other persons. Detection of any virus, including the novel coronavirus, in the tract raises a fundamental question. In what form does the virus exist in the tract? A virus such a parasite cannot exist on its own, cannot reproduce and mutate. It therefore must have a carrier. So that carrier does the virus use to persist in intestinal tract, but also throughout the body? Can they be bacteria or yeast or some other single cells or multi-cell organisms? Next slide, please. The claim that the HIV was transmitted in humans from monkeys about 100 years ago has not been sufficiently confirmed. The spread and incubation period and other symptoms of the Black Death have led to the theory that epidemics may have been caused by hemorrhagic virus. According to our result, we have concluded that the Yersinia pestis was an agent in epidemic along with HIV. Our considerations are strongly supported by CCR5 delta 33 mutation, which protects against HIV infection, and based on mathematical model, has been present in Caucasian population for over 2,000 years. The combination of these two agents led to the devastating Black Death epidemic. The plague led to the decrease in the number of HIV carriers and increased the number of CCR5 delta 33 mutations in Caucasian population to 10 or 15% in northern to 20%. Next slide, please. In sub-Saharan Africa, this epidemic and subsequent sanitation process didn't take place, what explains the absence of CCR5 delta 32 mutation. This results in high level of HIV genetic information in this population. A virus like the parasite is not full-featured biological form and thus hard to find. Its main weakness is that it's hosted by bacteria or by yeast. Bacteria or yeast are the complete biological form and they can be eliminated. But destroying microbes carrying the virus by antibiotics or other drugs, the virus ceases to exist. Thus many viral infections can be stopped, including novel coronavirus epidemic. We experimentally proved the presence of coronavirus in the rectal swab using RT-PCR on the cohort of very infected persons 4 weeks after infection. Next slide, please. And finally, about virus in general. Let's take a common one, the flu virus. He attacks people in autumn and winter. When he leaves, where? Will he travel to China or Hong Kong? How? By plane? And will be also buy a return ticket for October? It's not real, is it? No, he doesn't travel anywhere. He stays in the most of infected in the intestinal tract, hidden in carriers. It stays here until autumn and is under control of immune system. If the infected person's immune system is weakened, ordinary colds, illness, infection, injuries, the carrier of the virus multiplies, penetrates the organism of the infected person and can infect others. This cycle repeats itself every year, and most viruses behave similarly. Next slide, please. In some cases, of course, the virus completely leaves the body, but worldwide, the viral load in population is constantly increasing. We have many little about the intestinal tract. The proof of the presence of the virus hidden in carriers. It, it reveals the important and largest viral reservoir in our body. Virus in carriers settle in the intestinal tract, and after the failure of the immune system, they can subsequently pass into the body. Its accumulation continues because the new and new viral infections arrive. The viral load in intestinal tract is constantly increasing worldwide, which represents a biological time bomb for the human in the near future. By detection of the virus hidden in carriers was open gate. It's the first information about the danger localized in our intestinal tract. Next slide, please. It's assumed that the more components of the microbiome acting pathogenically participating in induction of wide variety of diseases will be discovered through concentrated research. Their number is difficult to estimate now, but I believe it's very large. Their detection and subsequent elimination will be an extremely difficult and long process because from individual to individual, based on my experimental experiences in oncology, virology, and genetics analysis and consideration, I can believe that the human organism is a perfect system that can repair its own error and disorders. But for me, this is an axiom. There is the existence of the life, of errors, damages, all kinds of disease, mutation, failure of individual organ are, in my opinion, induced by second kingdom in our body, the prokaryotic kingdom. Next slide, please. We have to live in symbiosis with it, I mean that prokaryotics. We cannot exist without it. But there have been a large changing here. The use of antibiotics, medicine, drug has created enormous pressure on the prokaryotic system. Sensitive microbes mostly didn't survive this pressure. However, the infinite variability, each bacterium is unique, allowed them to survive. Legally, one bacterium out of 10 million survive, creating a new clone and subsequently new generation disease resistance. This process is constantly repeated and new resistant forms are created. The original bacteria that live in symbiosis with organisms having required resistance lose the ability to a large extent. Their goal to survive forces them to disrespect the host. It should be remembered that they can act continually on a body 24 hours per day, maybe sometimes more. As long as organism rest, sleeps, they work, and if they have pathogenical potential, they continue attack it. Next slide, please. And so the original symbiosis gets new and new wreck. It's becoming weaker and more vulnerable. Due to contact with new form of potential pathogenic bacteria, the immune system is put under enormous pressure. The balance between these two kingdoms has weakened, is very fragile, and minor attack is enough to collapse it. How to stop this process? This must be our main goal for the future. If we do not damage to do it, the health status of the human population will deteriorate significantly. Thank you for your attention. I'm very hard in hearing after very much. Thank you. You can send questions to my mail address. Okay, thank you.
Thank you, Dr. Vladimir Zozak. That's a
Yeah,
wonderful presentation.
thank you for collaboration. Thank you very much. I'm
Thank
sorry.
you. Yeah, if anyone have any query, please email to Dr. Vladimir. He is available through mail. Let's move on to the next presentation. The next presentation is by Dr. Andrew A. Brunasio from SEAAB IR Research at Oral from Romania. Hello, Dr. Andrew. Are you ready with your presentation?
Yes, I am ready. First
Yeah.
of all, sorry for the delay with my presentation, but my connection
Okay.
was defective,
Yeah.
and so
Okay.
we
So
can start.
we are
Yes,
play. Yeah. Yeah. We are. I mean, like we are. We will share the screen from our end, and you can explain it. And please
yes,
instruct us with the next slide so that we are able to move the next slide.
yes, yes. My name is Andrei Bunaciu, and I'm senior researcher and the owner of this company, AAB underline year Research Limited. First of all, I want to thank the organizer for inviting me at this conference to present part of our scientific possibilities and for the help to connect us. My company is a young one being created in 2018 after my retirement. The field of interest of the company is related to the use of classical infrared spectroscopy in pharmaceutical, biomedical, and food analysis. Being a chemist, I like when I see my name written with chemical symbols. Next slide, please. Even we are not working in the same lab, our research group is composed of Professor Abulenein from Egypt and Professor Wang from Vietnam and me from Romania. But in time there were also other researchers who joined us in different projects. Next slide. As a result of our long collaboration with Professor Abulenein from 1992 and Professor Wang from 2011, we published in 2020 in Elsevier the book Vibrational Spectroscopy Application in Pharmaceutical, Biomedical and Food Sciences, where there is a part, the second one, next, dedicated to biomedical analysis. In this part, we present several important applications of vibrational spectroscopy, infrared and Raman techniques used in different infectious disease diagnosis. There are also, next slide, there are also some other reviews published by us in time related to infrared spectroscopy used in biomedical analysis. Next. The principal question of the present days is what are infectious diseases and how can we detect them earlier? The first part of the answer is simple for everybody, not only for specialists. Infectious diseases are readily transmitted from one person to another and are brought on by viruses, bacteria, fungus, or parasites. Regarding the detection, I will try to capture your attention with this presentation. Next slide. Infectious disease diagnosis using Fourier transform infrared spectroscopy when I can give a possible response. Next slide. From the early days of life on Earth, human and viruses coexisted, and there was an eternal fight between them in order to obtain the immunization and antiviral treatments on our side. Viral diseases are one of the key issues in public health. Nowadays, there are a great number of viruses in the environment, majority of them known by biologists, but there are others still unknown. Next. From historical point of view, Rhazes or Al-Razi from Persia was the first scientist in virology with his description of smallpox and measles, and he proposed the theory of acquired immunity. There are also other important researchers, next slide, in virology and immunology, such as Edward Jenner, considered the father of immunology, Pasteur, considered the father of bacteriology, and last but not least, Sir Frank Burnett in 20th century, all of them and many others having a great contribution to the development of this new science. Next. From the analytical point of view, we must underline the fact that the most used methods in clinical and hospital diagnosis are serological ones, very known for the clinical microbiologic specialists. These methods are based on detection of antibodies produced against the viruses. Once a certain antibody is detected, it means that a virus is present in the sample. The big problem of these techniques is that for viruses of the same family, cross-reaction can occur. Next slide. Let's talk about infrared spectroscopy. Next. Infrared is a type of radiation with wavelengths above 750 nanometers. It is divided into three regions: mid-infrared, near-infrared, and far-infrared. Mid-infrared, the most used, is used to study the fundamental vibration of a molecule. Near-infrared is sensitive to overtones and the combination of vibration in a molecule, while far-infrared it is used to study the rotation of a molecule. The next slide presents the general representation of an infrared spectrum, where we can observe the presence of peaks characteristic for each chemical bond. The region between 500 and 2000 centimeters -1 is called fingerprint region, being specific for each substance. There are a great number of substances in the world, but there are no two substances with the same infrared spectrum. Even the geometric isomer can be differentiated, so infrared spectroscopy can be used as identification tool. Taking into consideration, next slide, taking into consideration that we will talk about biological samples where water is the principal component, major component, not principal. It is important to see how looks a baseline spectrum, water and carbon dioxide. It is seen that around 3,400 centimeters -1 and 1,600, there are great peaks due to the water contribution. Next slide. The technique used for infrared are transmission, attenuated total reflectance, and transflection. For biomedical analysis, the simplest procedure is using attenuated total reflectance method. Next slide. The best interpretation of the spectra is performed taking into account the peaks observed, but sometimes it is difficult to establish the correct position of the peak, and then we must apply chemometric methods, PCR, this time principal component regression, and PLS partial least squares, or use derivative spectra. This slide presents the characteristic of a normal spectrum in bottom and the second derivative similar spectra, top. Next. We must underline the fact that there is a close correlation between the sample composition and the molecular spectral characteristic, and so for unknown sample spectra, it can be obtained a real prediction after several multivariate correction methods. Next slide. Infrared spectroscopy has the potential to serve as a diagnostic tool for detecting and discriminating different diseases due to the induced changes of chemical composition and structure. Next slide. In time were published many papers dedicated to this subject. Only in Science Direct there are more than 50,000 papers after research using infrared spectroscopy in infectious disease as keywords. There are, as can be seen, about 2,000 papers for this year, and more than that, about 50 papers for the next year. As I told before, next slide, the spectrum represents the sum of all the components present in a sample, even we are talking about major or minor components, and it is a fingerprint of that sample. Starting from the blood sample, it is preferable to use serum or plasma in order to reduce the number of components while each has characteristic peaks. Next, we will present some application of infrared spectroscopy in detection of some viruses that are the cause of infectious disease. The first experiments were dated around 1960, when for Lactobacilli species was obtained the first infrared spectra. Lactobacilli are not only beneficial organisms, but are among major isolates when advanced dental caries lesion and can act as causative agent in more serious infections. In 1990, next slide, Naumann and his co-workers established the principle of work for microbiological experiments using Staphylococcus aureus, a pathogenic bacteria which can produce suppurative infection or septicemia. Next, we will present other application for HIV. Next slide. This is a cause of pregnant woman infected with these viruses, that we can observe the difference between the patients and control spectra. Next similar spectra for other HIV infections. Next. And here it's a difference between control and AIDS patients and so on. Next. Typhoid fever is another. Next. And here is a infrared spectra for control and infected patients with typhoid fever. Next. For cirrhosis and next similar spectra. Next. Observe the difference between control and infected patients. Next. Uveitis. Next. Streptococcus that can cause pharyngitis, pneumonia, and other infections. Next. Hepatitis B and C. Next. Salmonella enterica with gastrointestinal disease. Next. Papillomavirus. Next. Escherichia coli. For this type of virus, next. Sharana evaluated the antibiotic resistance using infrared spectroscopy for cotrimoxazole drug. Of course, next, we cannot forget about SARS-CoV. Next, with its detection from blood. Next, swab. Next, and saliva, where the sample are obtained easier than in other cases. Next slide presents two infrared spectra for infection with SARS-CoV virus spectra at two different concentrations. The next two slides summarize some viruses detected using infrared spectroscopy, and the list can continue. Next slide is continue from this one. In conclusion, next, vibrational spectroscopy, infrared or Raman, has demonstrated significant diagnostic capabilities for microbiology, with early studies showing accurate identification of bacterial pathogens. Infrared offer advantages of simplicity, rapidity, low cost, and minimal sample preparation. Next, the greatest advantage of infrared spectroscopy is determined by the high molecular sensitivity. Next, the proposed method passes the problem associated with classical biochemical methods, which are expensive, time-consuming, and destructive. Next, it is Sometimes it is necessary to use chemometric methods for determination. Next. All of us fight for a better world, and this fight will be a great success if all of us has a proper word like mine. Next slide. They are my three grandchildren, Emma 5, Lucas 3, and Iris 1. For more details, next. Here are my contact data. And I want to announce next, I want to announce you that Molecules Journal prepare a special issue related to the use of infrared spectroscopy as a tool for analytical determination, where I am one of the guest editors, and we invite you to join us with interesting manuscripts. Next, I want to thank Professor Abulenein and Professor Huang for their support in realizing all our papers, and to all of you for your attention. Thank you very much.
Thank you, Dr. Andrew. That's a wonderful presentation. Does anyone have any query? Hope there are no queries. Let's move forward with the further presentation. The next presentation is by
Thank you
Dr.
very much.
Thank you, Doctor. The next presentation is by Dr. Karima Warda from Qadi Ayad University, Morocco. Hello, Dr. Karima. Are you ready with your presentation?
Hello, doctor. Yes, I'm ready.
Yes, so I'm making you as
Can
a
you hear me?
presenter. Yeah, I can hear you. I'm making you as a presenter. Kindly click on the share button to share your screen, please.
Yes, okay. Yes. Can you say my presentation?
Yes, your screen is visible. Please make it a full screen and start your presentation.
How can I make it full screen? How can I make it full screen, please?
Hello, Dr. Karima. Please make it a
Yeah.
full screen.
Yes, I don't know how.
In the bottom corner, there will be an icon for full screen. Can you please click on that? Hello, Dr. Karima.
Yes.
Yes, can you find? Okay. Please open your PPT.
Yes. It's opened from here. It's very small. I can't see where. It's very small.
Hello, Dr. Karima. Kindly
I
open your
tried.
PPT.
I tried to open it from go to. Yes. It's
kindly click on the PPT PowerPoint presentation down there in the bottom, at the bottom.
Which bottom? Share?
Please open your PPT.
Yes, okay. And I have notes. I have normal and
Yeah. Please move us beside that. Again,
mode flexion.
another icon beside that. The next icon beside that. Yeah. It's perfect. It's full screen now, and st-
Okay.
please close that. Hello, Dr. Karima. Again, open your PPT.
Please wait a moment. I ask for help. Présentation لو فين هي؟ واش هذا؟ لا البريزونطاسيون ديالي بيبيتي انا حلها تدخل البيرو فين هي؟ كنسبح اش كان مكتوب هنا؟ هادي دونك انا هنايا جوتو واش هذا؟ هذا ها هي صافي اكتيفي الميكرو ديالك راه ماكتيف هنا راه ماكتيف can you save my presentation بلاتي
Yes, your screen is visible. Please click on the same icon that you clicked before to make the full screen.
it's okay now it's okay now
Yes, it's perfect. Please continue with your presentation.
okay thank you can you hear me yes
Yes, you're audible.
yes thank you hello everyone I'm pleased to be among you today to present our work um, on the strain of Streptococcus pneumonia in Marrakesh region of Morocco. First, I apologize for my voice. I'm currently until with a cold, which has left its house. Thank you for attention and understanding. I will begin my presentation with an introduction, followed by objectives and methods of works, the results and discussion, and finally, a conclusion. Yes. Streptococcus pneumonia, also known as pneumococcus, is a gram-positive encapsulated bacterium identified by its lancet-shaped diplococcal morphology. Sorry for this. Uh, in 1920, it was classified as a new species and named Diplococcus pneumonia. Later, in 1975, it was classified as Streptococcus pneumonia due to its similarity with streptococci. Furthermore, pneumococcus can be distinguished from other oral streptococci by its frame-shaped capsulated diplococcal morphology, solubility in bile salts, and sensitivity to aptoxin. How does Streptococcus pneumonia affect the body? As an opportunistic inhabitant of nasopharynx, colonization of nasopharynx presents the preceding steps before infection occur. The host bacterium relationship can be into an imbalance, leading to either direct extension pneumonia from throughout the Eustachian's tube to middle ear or a sinusitis, or in the case of non-invasive pathology like middle ear infection, sinusitis, and pneumonia, or dissemination throughout hematogenous route for invasive pathology like bacteremia, meningitis, or osteoarthritis. This pathological situation depends on the host's decreased local and humoral defenses and the virulence of the microorganism. Pneumococcus is a most common cause of community-acquired bacterial meningitis in adults and one of two major agents responsible for ear infection. It's also the leading cause of community-acquired pneumonia and infectious lung disease. In children, invasive local infection primarily affects those under the age of 2 years and can lead to pneumonia and meningitis. The latter results in mortality in 8% to 50% of cases, along with frequent sequels such as epilepsy, deafness, and paralysis. The microorganism is responsible for little of 10% of death in children under 5 years of age, primarily in developing countries, posing a major public health issue. In addition to this, invasive infection, pneumococcus is responsible for a significant proportion of bacteria upper respiratory tract infection. At least 1/3 of bacterial middle ear infection in child are caused by this pathogen. While these local infections do not endanger in child's life, they are considerably more common and therefore lead to significant medical and social costs. Virus factor. The pathogenicity of pneumococcus is expressed throughout virulence factors, some of which are cytoplasmic, such as pneumolysin toxin, the capsule and capsular antigen. The antigenic characteristic of pneumococci is primarily done using the capsule, which is an immunogenic polysaccharide. This antigenic polysaccharide forms the basis for the serotypic classification of pneumococci. 101 pneumococcal serotypes are now known, all potentially pathogenic to human. However, A small number of them are more violent than others, and about 10 serotypes are found in all invasive infection. These serotypes include 1, 3, 5, 6, 14, 19, 23, 4, 9V, and 18C. From one serotype to another, the primary structure of the polysaccharide chain vary in terms of number, size, sequence, type of band, and the presence or no eosological substituent. Each serotype has an antigenic formula comprising between 1 to 5 different antigens. Role of the capsule. The capsule is gelatinous outer layer produced by bacteria to protect against phagocytosis and other external threats. In addition to being a common feature in almost all clinical isolates for pneumococcus, the capsule is also a characteristic shared by most extracellular pathogenic bacteria. Non-encapsulated pneumococci can colonize the nasopharynx, and due to selective pressure from current vaccines, they are considered an emerging population. Non-encapsulated pneumococci can cause a localized infections such as sinusitis, conjunctivitis, keratitis, and middle ear infection. This table shares another factor of virulence like a surface under this function, like a surface protein, CBCP and lipoprotein, pneumolysine, autolysine, immunoglobulin A protease, and hydrogen peroxidase. Those are also virulence factors for pneumococci. Objectives. For one information, because the capsule is very abundant and exposed to the surface, and antibody directed against the capsule are generally highly opsonic and protective against invasive disease. Currently, the capsule is used as the primary antigen in several vaccines against pathogen. Yes. Objectives of our study. First of all, objectives of our study was to determine the colonization and carry rates of Streptococcus pneumoniae in healthy children under 5 years old. We aimed to establish the distribution of the different serotypes based on age group and various regions studied. Additionally, we evaluated the resistance of these isolate strains to different antibiotics used in the treatment of invasive and non-invasive pneumococcal infection. Study characteristics. Our study was prospective and took place from November 28 to February 20 and 22. Study population consisted to healthy children aged 6 to 60 months who had not received antibiotic treatment in the past 7 days. Prior the sample collected, parental consent was obtained. socio-demographic and socio-demographic information, vaccination status, and antibiotic consumption in the 3 months preceding the sampling were recorded on a data sheet. Material and method. Study design. This prospective study was conducted in Public Health Center in Marrakesh, Morocco. Healthy symptomatic children aged less than 5 years old visiting public health center for vaccination were randomly selected. A healthy child was defined as a child presenting with no fever, no signs of respiratory infection, and no antibiotics consumption during the last 7 days. A questionnaire containing demographic, socioeconomic, and clinical data was completed. The collection of nasopharyngeal specimens was performed using the sterilized flocked nylon swab placed in a medium containing skim milk, tryptophan, glucose, and glycerol. One nasopharyngeal swab was collected from each child. Swabs were sent to the Microbiology Virology Laboratory of Faculty of Medicine and Pharmacy in Marrakesh, Morocco. Only child with pneumoniae positive culture were required in this study. So the ethical committee of University Hospital Center of Mohammed VI, Marrakesh, Morocco, approved this study and gave us the permission to conduct this study. For identification of pneumonia, the isolate of nasopharyngeal specimens were initially cultured on cholesterol nalidixic acid agar supplemented with 50% blood and incubated overnight at 37 degrees at 5% of CO2 atmosphere. Pneumoniae isolates were identified based on typical colony morphology, alpha hemolysis, gram-positive staining, negative catalase reaction, optogen susceptibility, and bi-susceptibility, and an agglutination test slide pneumokit. The strains of S. pneumoniae colony were then transferred into brain heart infusion baths, supplemented with 50% glycerol and kept under sorry for that. And frozen. Antimicrobial sensitivity testing. Antimicrobial sensitivity was tested on Muller-Henton agar supplemented with 5% sheep blood using the desk diffusion method. Beta-lactamase resistant is in Streptococcus pneumoniae was firstly determined using an oxacillin disk with concentration 1 microgram according to the ECAST recommendation. According to the ECAST recommendations. And state for strains with an oxacillin zone diameter 2 milligram per liter, respectively for amoxicillin and for under 0.5 milligram per milliliter for and 0.575 to 2 milligram per liter and superior to 2 milligram per liter, respectively for ceftriaxone. Antimicrobial susceptibility was also tested against norfloxacin, gentamicin, vancomycin, erythromycin, clindamycin, pristinamycin, tetracycline, chloramphenicol, and CXT. In case of a pneumonia strain was resistant to erythromycin, the testing was performed to detect the following phenotype: MLS inductible phenotype or MLSB constitutive phenotype or M phenotype. And for serotyping, the detection of S pneumonia serogroup was performed using Emulix pneumotest agglutination test. The serotype was performed using real-time polymerase chain reaction, following the recommendations published by the Center of Disease Control and Prevention (CDC). Killing reaction was accomplished for serotyping serogroups 9, 6, and 23rd. Finally, statistical analysis data were performed using a species/PC 23 program. Participant characteristics were expressed by count and percentages, or median and interquality range. The key to test was done to compare the non-susceptibility of oxacillin positive and oxacillin negative strain to other antibiotics. A p-value under 0.05 was considered statistically significant. Demographics data. Out of the 645 children, the overall average age was 22.0 months, 22.5 months for girls, and 22.98 months for boys. 67.3 of children carrying S pneumonia were between the age from 6 and 24 months. The age group between 1 and 2 years was predominant, representing 277 children, or 43%. Regarding breastfeeding, can you hear me? Hello?
Yes, you're audible.
Yeah.
Hello, Dr. Karima. You're not audible. Can you please check your microphone settings?
Can you hear me now?
Yes, yes, you're audible now.
Thank you. I was in, yes, regarding breastfeeding, 5-6% of children were breastfeeding, while 28% were not. 8% for these children were primarily carried for at home, while 20% were in daycare. 65% of these children has more than one sibling, while 35% did not. In terms of antibiotics therapy and vaccination in our study, 28% of child has taken antibiotics within the third month preceding this study, while 55% 65% has completed their vaccination program, compared to 35%. Among this study variable characteristic, only breastfeeding, having more than one sibling at home, and taking antibiotics within the 3 months period to sampling has a significant effect on the healthy carriage of Streptococcus pneumoniae in the child included in this study. Regarding the carriage in our study, 25% of sampled child were carrier of pneumococcus. Carriage has significantly decreased compared to our study before introduction of vaccine, which was uh, which was uh, which was 35%, which was 45%. It's also lower compared to study conducted in our country in invasive strains, which have carried a rate of 14.5%. Regarding the distribution of serotypes among the vaccine serotype, regarding the distribution of serotype among the vaccine serotypes, we observe the dominance of serotype 14. You see here in our currently study, and followed by serotype 3 and 6A, which accounts for 60.8%. There is also a decrease in serotype 20. Followed by serotype 3 and 6A, which account for 16.8. There is also a decrease in serotype in 19A and 19F, and an increase in non-stable serotypes. Comparing this distribution compares this distribution with previously study, we noticed that an increase in the prevalence of serotype 14, which has risen from 7 to 16.8% in 2020. There is also a significant decrease in serotype 19A and 19F, which have decreased from 6.74% and 11.65% in 2009 to 0.7 for both in 2022. We also observed the dispersion of serotype 4 in 2022 and the appearance of serotype 3 in the same years. Therefore, in 2022, we can see the dispersion of some vaccine serotype and the emergence of many non-vaccine serotypes, such as 9, 8, 11, 15A and F, 15B, and 23B. The emergence of serotype 3 might pose a public health concern as it requires a significant amount of antibody for effectiveness opsonization and subsequent protection of the patient. Unfortunately, this amount is not triggered by a current conjugate vaccine. If we compare the distribution of serotype between 2021 and 2022, we notice that the predominance of vaccine serotype 14 in 2021 has decreased in 2022. Vaccine serotype 3 included in PVC 13 remain predominant among children in 2022. Some vaccine serotypes are gradually increasing 50%, 20%, 25% in 2021 compared to 27.5% in 2022. Non-vaccine serotypes such as 15A and F, 6A and 9A are emerging. Many studies have focused on the fluctuation of pneumococcal serotype and the impact of vaccination on their distribution. They have reported that under vaccine selection procedures, there is pressure. There is a lot of change in the epidemiology of serotype responsible for invasive infection. Almost all of them have reported the phenomenon of serotype replacement. Antibiotic resistance. All pneumococcal strains isolated from healthy child in Marrakesh were sensitive to norfloxacin, gentamicin, chloramphenicol, and vancomycin. The rates of multi-drug resistant strain decreased from 43.33% on 29 to 27% in 2022. We're comparing the years 2021 and 2022. The following trends were observed. Decrease in the frequency of strains with a reduced sensitivity to penicillin, 51.5% in 2022 compared to 62.7% in 2021. The frequency of resistance to amoxicillin was 26%, amount The strains were resistant to penicillin. These strains were classified as lower level resistant to ceftriaxone. We observed an increase in frequency of pneumococcal, strains resistant to erythromycin, vancomycin, and tetracycline. In a systematic study, it's observed that pneumococcus ranked among the organism with significant rise of multi-drug resistance after E. coli, Staphylococcus aureus, and Klebsiella pneumoniae in 2022. A second study, Vietnamian reported a general trend in increased resistance of pneumococcal strains between 1999 and 2013 to penicillin, ceftriaxone, tetracycline, erythromycin, and CXT. To conclude, this study highlights a notable decrease in pneumococcal carriage and an increase in non-vaccine serotype strains among healthy children. Antimicrobial resistance has been observed in several commonly used antimicrobials, including beta-lactamase and macrolides. Ongoing surveillance is essential to monitor and control the epidemiology of pneumococcal carriage and resistance in Morocco. And thank you for attention. And I'm very sorry for this presentation. I'm very, very tired because of my cold. I'm sorry.
Thank you, Dr. Karima. That's a wonderful presentation. Does anyone have any
Thank you.
query? Hope there are no queries. Thank you once again, Dr. Karima, for your wonderful presentation.
Thank you for you, and I'm very sorry for my voice and my presentation. I'm very
We can
sorry.
It's okay. We can understand your condition. And as per the schedule, let's take lunch break. We'll get back to you after 30 minutes from now. Kindly stay connected.
This conference will now be recorded.
I hope everyone is back for the afternoon session. Now let's start. Our first presentation is from Dr. Carola Montenico from University de Americas, Chile. Hello, Dr. Carola. Are you ready with your presentation? Hello,
Yes,
Dr.
it's correct.
Carola. Okay, now I'm making you as a presenter. Kindly click on the screen button to share your screen, please.
Okay, thank you. Voy a compartir.
Please open your presentation. Can you make it as a full screen? Okay, it's perfect. You can start your presentation.
Can you see?
Yes, it is visible. You can start your presentation.
Okay. Um, Thank you very much for the invitation. My name is Carola Montesino. I'm from Chile. Their research is evaluation of the effectiveness of quarantines measures during the COVID-19 pandemic in Chile using Bayesian structural times series models. The authors are Claudia Barría, Guillermo Ferreira, Bernardo Lagos, and Carola Montesino. We know that the pandemic started in Wuhan, China, and the WHO declared COVID-19 a pandemic on March 2020. Quarantines have been an effective epidemiological strategy to block the transmission of infectious disease in human history. The term quarantine has been applied as self-quarantines, for example, in the home, and mass quarantine in cities and regions and countries. One of the goals of mass quarantine is to flatten the spread curve of the projected number of people who could become infected over a period of time. The main objective of this study is to analyze the effectiveness of national and regional quarantine in Chile during the pandemic, based on information published by the Ministry of Health of Chile. And the question is, were quarantine measures effective in Chile during the COVID-19 pandemic? Methodology. Quantitative research. We used incidence, prevalence, and new cases of COVID-19 in phase 1 and in phase 1 and phase 2 quarantines during 2020, from March 2020 to March 2021. And we used statistical tests, Bayesian structural times, series model to project and model the curve and infection. And results. This is Chile. Chile is in Latin America, and we have 16 regions, north, center, and south. And in this chart, we can see quarantine phase 1. Phase 1 is total insulation, and the black curve is new cases in the regions. And red curve is quarantine phase 1. And in the north, first region, we can see new cases were decrease when with quarantines, decreasing, decrease. And the south, total insulation, new cases were increasing. And the capital is different because we have new cases, and with the quarantines, the new cases were decreasing. And phase 2, more mobility. Black curve is new cases, and yellow line or yellow curve is quarantine phase 2, more mobility. In the north, new cases were decreasing here, decreasing. And the south, the new cases were increasing too, like phase 1, increasing. And the capital in the center here, the new cases were decreasing with phase 2. In this chart, we can see infection in all regions. Magenta line is infection and green line is decrease of infection. In this point is March in the region, and this point is December 2020. And we can see more infection in March in all regions. And the green line is decreased infection, and quarantine is effective. Results. Phase 1 quarantines were more effective in the north, for example, Arica, Antofagasta, Atacama, Coquimbo, in the north, here in the north, because new cases were decreasing with quarantines, quarantine phase 1 or quarantine phase 2. But the quarantines implemented in Central South and extreme South Chile, region 8, 9, 10, 11, 12, 14, 16 were not effective because the new cases were increasing with quarantines, quarantine phase 2 or phase 1 in total installation increasing. And the capital is different, is a special case, because in capital, here in the center, here, the new cases were decreasing in phase 2, but the quarantines in the capital were more restrictive than other regions. And the water in north is warm, and the south, the water is cold, and the center we have Mediterranean climate. As description has conclusion, as description, similar strategies used in other countries in the world and Latin America. Has description the places where quarantines were conducted early and timely manner experienced low health and economic cost, and additionally the implementation of prevention and epidemiological control measures ensured the effectiveness of the quarantines. As conclusion, it would be relevant in subsequent studies to identify the sociocultural variables that favored the increase in infection during the first quarter of 2021, and to analyze the behavior of the effective numbers of reproduction with the start of the vaccination plan against COVID-19. And this is the reference. Thank you for your attention. My mail, and this research is here, and this is the journal. Is open access. Thank you very much for your attention.
Thank you, Dr. Carola, for your wonderful presentation. Does anyone have any query? Hope there are no queries. Let's move forward with the further presentations. The next presentation is by Dr. Syed H. Hogg from Born Secure Mercy Health, St. Rita's Medical Center from USA. Hello, Dr. Syed. Are you ready with your presentation? Hello, Dr. Syed. Are you ready with your presentation? Hello, Dr. Syed. Are you ready with your presentation? The next presentation is by Dr. Syed H. Haq. Hello, Dr. Syed. Are you ready with your presentation? Hello, everyone. I hope there is some technical issue with Dr. Syed. So let's move on to the next presentation. The next presentation is by Dr. Rayad Jab from Mayo Clinic Health System from United States. Hello, Dr. Rayad. Are you ready with your presentation?
Hello, yeah, I am ready actually.
Okay, so I'm making you as a presenter. Please click on the share button to share your screen, please.
So for sharing, I have three options: Microsoft Edge tabs, Windows, and entire screen.
Yeah, please click on the entire screen option. Now open your PPT. Yeah, please make it a full. Yeah, it's perfect. Please continue with your presentation.
Okay. Hello everyone. I'm presenting a case about hepatic actinomycosis that's mimicking malignancy. My name is Raid Jaber. I am assistant professor at Mayo Clinical System in Eau Claire, Wisconsin. So the history of this patient is a 73-year-old white male patient. He presented with chief complaint of vomiting up blood, hematemesis. His past medical history is significant for stage 3 chronic kidney disease, heart failure with reduced ejection fraction, and he had history of hemorrhagic cholecystitis and underwent ERCP and biliary stent placement. That was a year prior to his presentation, more than a year, 15 months prior to his presentation. His social history is significant for 20 pack years smoking history. He quit 2 years prior to admission to the hospital. So in presentation, he underwent CT abdomen pelvis with contrast that showed 9 centimeter liver mass that's invading the gastric mucosa, concerning for cholangiocarcinoma or hepatocellular carcinoma. And here you can see on initial CT abdomen, this is the right lobe of the liver with 9 centimeter liver mass, and here is the same liver mass. And it is invading the stomach here. This is the stomach. So with his presentation of hematemesis, he underwent upper GI endoscopy, and that showed tumor at the curvature of the gastric antrum with central ulceration, as we can see here. And this is demonstration of his mass that's invading the gastric antrum. So he underwent clipping to control that bleeding. That was the presumed cause for his upper GI bleeding. So he underwent clipping for that reason. With his liver mass seen in his CT abdomen pelvis, so he underwent liver biopsy. That didn't show any evidence of malignancy and the histopathology. But gram staining of the sample showed gram-positive rod that was consistent with actinomyces species. So here at this point there was diagnosis of hepatic actinomycosis, and here we can see in the histopathology there are multiple colonies of gram-positive rod or bacteria. Also, despite being asymptomatic from respiratory standpoint, on the upper cuts of his CT abdomen, there was moderate right-sided pleural effusion, and there was bilateral nodular interstitial infiltrates and 1.8 centimeter left lung base nodule. As we can see here, the moderate to large right-sided pleural effusion. And this is the 1.8 centimeter left lung nodule. So the hospital course general surgery team was consulted. Patient wasn't a surgical candidate because of the direct invasion of that liver mass to the stomach. So he will be at high risk for GI bleeding. So no surgery was done for him. With his CT chest findings, he underwent thoracentesis that was consistent with exudative pleural effusion. He had negative pleural fluid cultures, including bacterial, fungal, and mycobacterial cultures. And we sent also the broad-range bacterial PCR from that sample, from the pleural fluid. The 16S came back negative. He underwent bronchoscopy also that was negative for the bronchoalveolar lavage, negative for galactomannan, AFP smear, gram stain, and cultures. So at that point, given that no surgical intervention is warranted and there is diagnosis of hepatic actinomycosis, the patient was discharged for on IV penicillin, continuous infusion for that diagnosis, for the actinomycosis. So 6 weeks after starting IV antibiotic with penicillin, his repeat CT abdomen showed decreased size of the liver mass from 9 centimeter initially on presentation to 5.2 centimeter. So at that point, we switched IV to oral penicillin, 500 milligram every 8 hours. 6 months after treating with oral penicillin, his repeat CT, chest, abdomen, and pelvis showed complete resolution of that liver mass, as we can see here on his CT abdomen, and also improvement of his bilateral pleural effusion and bilateral infiltrates. So I'm gonna talk in a few slides about actinomycosis in general, then hepatic actinomycosis specifically. So actinomycetes are commensal inhabitants of the oral cavity and GI tract. Usually they present with polymicrobial infection. It's not uncommon to have polymicrobial infection on cases of actinomycosis. And actinomyces species secrete proteolytic enzymes, and that's make it prone to cause cutaneous fistula and directly invade surrounding tissues. So it can present subacutely as granulomatous inflammation, abscess with direct spread to adjacent tissues, or sinus tract or fistula. The most common site of infection is the cervicofacial area. in 50% of cases, according to a study by Wong and her colleagues. That was followed by abdominal involvement in 20% and thoracic involvement in 15 to 20%. The most common site in cases of abdominal actinomycosis were the appendix, then the ileocecal region. And in these abdominal cases, only 15% of cases were hepatic actinomycosis, and that accounts for 5% of all actinomycosis cases. So it's not a common entity. That's why it's difficult to have the diagnosis. So this is a review of literature with a systematic review of literature of 64 cases of hepatic actinomycosis that revealed that 64% of cases were male, and that's consistent with other actinomycosis on other sites. And that was of unknown, there was no clear explanation why male are more prone to get that infection. 61% were primary, not disseminated cases. The majority of cases, or at least 60% of cases, were primary. 92% were immune competent. Around 26%, one-fourth of cases were polymicrobial infection. And 80% had identifiable risk factor, and that's most commonly was prior history of abdominal surgery, foreign body in the abdominal pelvic area like intrauterine device or biliary stents, and then diabetes or immune suppression also. So here we can see about risk factors for hepatic actinomycosis. That includes abdominal surgery was the most common one, and that's followed by foreign bodies, either intrauterine device or biliary stent. Diabetes mellitus in around 17% of cases, smoking, hypertension, alcohol consumption, oral disease, chronic pancreatitis, or renal failure. So clinical manifestations, onset was chronic or subacute with extended duration of symptoms. The mean duration of symptoms was 3.1 months. Most common symptom was abdominal pain in around 2/3 of cases, fever 62% of cases, and weight loss in 48% of cases. Initial workup was significant for leukocytosis in 2/3 of cases, anemia 55%, elevated alkaline phosphatase 45%, and they found that less than 1/3 of cases have elevated AST and ALT levels, and that's unlike pyogenic liver abscesses. In these cases of pyogenic liver abscesses, at least 2/3 of cases will have elevated liver enzymes. So that's something you can consider when you are dealing with, when you are considering diagnosis of hepatic actinomycosis. The diagnosis is usually confirmed by histopathologic examination of different specimens, and that happened in 67% of cases in this case series. Culture and histopathological examination in 5 cases, 8%, and culture alone diagnosed the hepatic actinomycosis in 7 patients, which accounts for 11% of cases. And here we can see that out of 40 patients whom cultures were obtained for them, only 18%, less than 50%, came back positive. So that was attributed most likely due to prior antibiotic therapy or inadequate culture condition, because this culture should be sent to the lab within 72 hours of getting the sample, and shouldn't be refrigerated or frozen. So if it is refrigerated for any reason, it should be discarded. Again, the modes of diagnosis here, the most common is histopathological examination. And among these, 67% of the histopathological examination, the majority were either liver biopsy, liver puncture biopsy, or liver lobe resection. And here we can see the culture account for 11%, pathology and culture both in 8% of cases. So regarding imaging with CT, MRI, or ultrasound did not give a definitive diagnosis for any of the patients because these modalities cannot differentiate between abscess, I mean biogenic liver abscess, hepatic actinomycosis, and sometimes can be misdiagnosed as malignancy. And here, if we go back to this diagram, we can see that liver lobe resection happened in 30% of cases, and most of these cases were initially diagnosed as cancer. So that's why they proceeded for surgery with liver lobe resection. The most frequent finding was single abscess or mass in this imaging. Most commonly in the right lobe, and 52%, in left lobe was 26%, and on both was 21%. And there was suspicion of liver malignancy or mets in 50% of cases in this case series. Like more than 30 cases were initially suspected. There was suspicion of liver malignancy. Treatment modalities in this case series were surgery or drainage and antibiotic in 44 cases, accounting 68%. Antibiotic alone in 18 cases, accounting for 28%. Liver lobe resection alone in one patient. There was no information about treatment in one case, and one of the patients died before starting the treatment. The antibiotic selection, most commonly used antibiotics were penicillin, amoxicillin, or doxycycline and ampicillin. So all of them are penicillin derivatives, which we all know that it's the first-line therapy for actinomycosis. The mean duration of treatment was 5.5 months, ranging from 1 week to 24 months. So take-home messages from this case that hepatic actinomycosis is a difficult disease to diagnose, and that's because of its rarity. It's not a common entity. Non-specific presentation, imaging cannot differentiate, and culture is usually negative, or it's negative in more than 50% of cases, according to this case series. In which cases we should suspect hepatic actinomycosis in patients with liver mass who presented with also poor oral hygiene, history of IUD or biliary stent, or prior intra-abdominal surgery. This infection is confused with malignancy, which may lead to extensive surgery. And in many patients, infection can be controlled using antibiotics, as in our patient here. He was treated with at least 6 weeks of oral penicillin, and there was complete resolution of his liver mass. Tendency of liver disease to recur. So there is tendency of this entity to recur, so prolonged duration of antibiotic is warranted. Thank you, and I'm happy to take any questions.
Thank you, Dr. Raid, for your wonderful presentation. Does anyone have any query?
No, thank you.
Hope there are no queries. Let's move forward with the further presentation. Once again, thank you, Dr. Raid, for your wonderful presentation. And the next presentation is by Dr. Reda H. Mitani and Dr. Farid Gargas from Kingston Hospital NHS Foundation Trust from United Kingdom. Hello, Dr. Reda and Dr. Farid. Are you ready with your presentation?
Yes, we are.
Hi.
Thank you.
Yes.
Okay, so I'm making Dr. Farid as a presenter.
Yes, please.
Yes, please.
Yeah, please click on the share button to share your screen, please.
Is it visible now?
Yeah, please open your PPT. Make it a full screen.
Can you see it now?
Yeah, it's perfect. Please continue with your presentation.
Hi, my name is Farid Girgis. I'm a surgical registrar in Kingston Hospital. We are presenting this case series with my colleague Reda Methani, but we have a bigger team helped collecting this data. This case series about invasive group A streptococcal infection in emergency surgery. We collected this case series in our hospital in Kingston Hospital. So who are we? We are in Kingston Hospital. That's it's southwest London, and we are a single-sited hospital, but we work jointly with many hospitals around us. And we serve approximately 300,000 people locally around us. And here you can see our values of our hospital. So the content of this presentation, first I'll speak about introduction about strep A invasive infection, then about the objectives of this project, then statistics, methodology, results and outcomes, then conclusion and the take-home messages. So as for the introduction, the strep A infection can be a challenging infection if presenting as invasive infection in acutely unwell patients. And it can occur both in invasive or non-invasive way. And the invasive infection is defined as isolation from infection in normally sterile areas of the body, like the blood, or non-sterile sites in case of septic shock. Historically, Group A streptococcal infection caused major epidemics, but advancements in hygienic practices and modern medicine led to decline in these severe infections. However, there was rise in the prevalence of invasive gas infections, especially after the post-COVID emergency phase. It's well known that conditions like necrotizing fasciitis or myositis can be fatal and it can reach up to 100% mortality in myositis. So early detection of invasive gas is very crucial, and treatment should involve multidisciplinary team, sepsis 6 protocol, and surgical debridement if needed. So here are the objectives of our project. Because gas is fatal, so we did this project to highlight the rising incidence in invasive gas, especially after COVID pandemic, to emphasize the significance and the importance of considering IGAS as a differential diagnosis in unwell emergency surgical patients, and to provide insights on management strategies. Here, this schedule highlights the statistics of gas infection nationally in UK. And you can see the highest point of statistics was in 2022 and 2023. And there has been multiple explanations. If you scan this QR code, you will read the full report about that and the explanation, but it has been explained. It might be because of decreased exposure during the pandemic to gas, and people went again after, during the pandemic, there was no exposure to gas, and people then got exposure. So that led to increased incidence of invasive gas. And here's the WHO records as well across Europe. There have been reports from multi-countries, increased incidence as well in scarlet fever and invasive gas. That report was in December 2022. So the methodology of our case series. So you can see here that we did the data collection from December 2022 till August 2023, and we gathered our data retrospectively from electronic records. And the inclusion criteria, we included all the patients testing positive for gas isolated from sterile sites or from non-sterile sites in case of septic shock, and that's as per definition for IGAS. So our results at this period, we collected 6 patients were identified, 4 females and 2 males. The average age was 55. So 4 of them had necrotizing skin infections, and me myself have been involved in a few of these patients for debridement. And 1 had bacterial peritonitis, and 1 had streptococcal bacteremia. So all of them presented in septic shock. They were managed by a multidisciplinary team according to sepsis 6 protocol. 5 of them had immediate surgery for debridement. So as for lab results and diagnosis, 5 of them were diagnosed by culture, whether blood, tissue, or wound culture, and 1 via genital swab. 50% of these cases were sensitive to both clindamycin and amoxicillin, and it's very important at this point because that's considered as the first line in treatment of IGAS, and the rest of them were sensitive to 1 of them. Of course, on presentation, as I mentioned before, all of them presented with septic shock, and all of them had markedly raised inflammatory markers. They had low albumin, which was expected because of sepsis and acute kidney injury. As for the outcomes, on the left-hand side, 3 patients were transferred to tertiary centers once stabilized and had the initial management. 1 of them transferred for neurosurgical input. That was the patient who presented with bacterial peritonitis because she had ventriculoperitoneal shunt. So she had to be managed under 1 of our tertiary centers under neurosurgical team and the general surgeons. And 2 patients transferred for plastic grafting. On the right-hand side, 1 discharged for community follow-up dressing, 1 discharged home, and 1 unfortunately died because of streptococcal toxic shock syndrome. So the conclusion and the take-home messages here, that we have to highlight that it's crucial for the frontline doctors to consider eye gas as a differential when they deal with critically ill patients, and early aggressive approach should be considered by simultaneous systematic resuscitation, multidisciplinary team, surgical intervention when needed, tailored antibiotic therapy, immunoglobulins if needed. That's to minimize morbidity and mortality. These are the references. Thank you. I'm happy to receive if any queries or comments. Thank you.
Thank you, Dr. Farid Gerges, for your wonderful presentation. Does anyone have any query? Hope there are no queries. Thank you once again, Dr. Farid Gerges, for your wonderful presentation.
Thank you.
And so, so should I make Dr. Reda the presenter now?
I'm not too sure.
Hello, Dr. Reda Mitani. Are you ready with your presentation?
That's our only presentation regarding this case series, but I'm checking if Dr. Reda has any comments. But we prepared this presentation together.
Okay.
I think we are fine now. Thank you.
Okay, thank you so much. We'll move further with the presentation. So the next presentation is by Dr. Syed H. Hogg, DO from Bon Secure Mercy Health St. Rita's Medical Center from United States. Hello, Dr. Syed. Are you ready with your presentation?
Yes, I am. Can you guys hear me?
Yeah.
Okay.
And so I'm making you as a presenter. Please click on the share button to share your screen, please.
Okay. Are you guys able to see my screen?
Yeah, your screen is visible. Please make it a
Oh,
full screen. Yeah, it's
Yes, we
perfect.
can
Please,
see it.
yeah, please
all
continue
right.
with your presentation.
Sure. So my case here is actually an interesting case about neurocysticercosis here in Lima, Ohio. A case titled "To Intervene or Not to Intervene," a case of symptomatic neurocysticercosis complicated by ventriculitis. Just to start off, there's no disclosures, no conflict of interest. So I'm going to start with the case presentation. We actually had a 24-year-old female. She was her only past medical history was honestly morbid obesity and some anxiety, depression, who came into the ED, altered mental status, and was unresponsive and having fevers. History was provided by her husband, and he stated that she had flu-like symptoms roughly 5 days ago that progressively worsened. These symptoms started with fevers, chills, and then progressed to nausea and vomiting with headaches. He found her minimally responsive with sonorous respirations, and that prompted him to bring her to the ED. He denied any other prodromes. He denied any sick contacts in the household, no changes in diet, medications, or recent environment or recent travels. The past medical history, like I said, was really non-significant in her case. She wasn't on anything else that would be a confounding factor for like a metabolic encephalitic process. So on her initial evaluation, She was responsive to stimuli, opening her eyes, tracking. She was able to move her extremities, but she was not conversing. She was found to be clenching her jaw. This was followed by beats of inducible clonus in both extremities on the lower side. And she also had a plantar reflex upgoing bilaterally, but no nuchal rigidity was appreciated. Her vitals were significant for a fever. She had a fever temperature of 104.3 Fahrenheit, and she was hypertensive. She also had a leukocytosis, and as a routine, they started with imaging in her, given the fact that she was young and she was presenting with ultramental status. Her initial CTA head and neck were negative for any intracranial pathology. However, the MRI demonstrated innumerable ring-enhancing lesions. You can see here the image on the top is the AX, then we have GRE and flare. So what was going on? We were not entirely sure. The initial concern was septic emboli, given her age factor and possibility of drug abuse or something like that. That, along with the fact that there was cerebral edema noted on the MRI, mass effect on the basal ganglia, thalami, and then her lumbar puncture, which resulted in elevated white blood count, low glucose, and a high opening pressure, all kind of pointed towards maybe bacterial meningitis, maybe from a septic emboli. In any case, she was pan-cultured, and she was started on empiric antibiotics with vancomycin, zosyn, as well as antiviral coverage given her age and risk factors. She was also started on Keppra for seizure prophylaxis because in her history she had some remote history of seizures, however, was not on any antiepileptics. Day 3, her MRI actually shows that there is improvement, however, now there's vasogenic edema. Unclear why there's this edema, but regardless, the emboli burden or whatever that was has actually shown improvement. But we still don't have a definitive source of what happened. Day 4, there's a rapid deterioration in her status. She is intubated for imminent respiratory failure, airway protection. The question is she having worsening cerebral edema, or is there subclinical seizures going on? There were some reported cases of yes, she was jerking and she had loss of consciousness. It was unclear what was going on. Regardless, she was transferred to the ICU, and the following consultants were consulted. At the time, infectious disease recommended continuing the antibiotics, antivirals until we can get better culture sensitivities on what's going on. Neurology was also consulted and recommended transfer to a tertiary center for continuous EEG to ensure that there's no underlying seizure or status going on. However, due to COVID-19 at the time, there was limited resources and limited beds, and we were not able to transfer her for this reason. So she had to stay with us. Neurosurgery was consulted as well, and they recommend no surgical intervention at the time, and in fact advised hypertonic saline and steroids to help with the cerebral edema. So our workup continues. We finally get a little bit of results back from our cultures. Blood cultures were negative. She did have a strep agalactic pneumonia. CSF cultures for common etiologies, bacterial, viral, HIV were all negative. Some zebras as well, toxoplasmosis, Legionella, Bartonella were negative. We had ordered HSV and nursisticosis, but they hadn't come back yet. Again, these were low on our suspicion given the fact of where we were regionally. Antibiotics were readjusted for the strep to Rocephin and ampicillin, high dose in case that there was bacterial meningitis going on, and the acyclovir was continued. EEG showed epileptogenic seizure tendencies, but nothing out of the ordinary other than that, and the TEE was negative for vegetations. So the question was, where are we now? What's going on? So, still with no definitive source for the septic emboli, we reached out to neurosurgery and asked, okay, we may need a diagnostic tissue now. So they reconsidered and were okay with the right temporal parietal craniotomy with biopsy. And the biopsy is here on the left on the histopath slide, as you can see. It didn't show any organisms or any mass or anything like that, but just showed reactive gliosis, so essentially didn't give us any results. After the biopsy, the patient was witnessed to have 2 focal seizures with myoclonus of the left upper extremity and effectively started on propofol and Vimpat. Finally, we get a break in our case. Serology comes back positive for neurocysticosis. In order to ensure it's not disseminated or anything of that nature, we go ahead and do the full workup. Stool sample was negative for any ova and parasite. We do an ophthalmologic exam to ensure that there's no cysticercosis of the retina, and there were none. So we start albendazole and praziquantel. Steroids now have been changed from our pneumonia coverage, lower dose steroids, to Decadron 0.1 mg per kg per day. And we notice an mild neurologic improvement within a week. She's effectively extubated, and her follow-up CT shows that there's improvement in her cyst burden, cerebral edema, allowing us to discontinue her supportive measure. The patient is transferred out of ICU to rehab and eventually in an extended care facility for further rehabilitation. So the question is, where did this all come from? As mentioned, this region is not very endemic. This is a white Caucasian female, so she's not from an immigrant family. She had no recent travels. No one in her household was sick. The hypothesis was, and the family did endorse, that the patient would procure her own pork rind. They were farmers. And she was seasoned into chorizos, maybe a possible source. Unfortunately, she re-arrived 2 weeks later to the ED with worsening mentation, increased work of breathing. Again, she was febrile with 104.7 Fahrenheit. She was tachypnic with a heart rate, with a respiratory rate in the 40s, 50s, and now she had significant leukocytosis. Her lactic acid was 3.7, procal was 0.8, CRP was 12.12, and this is after having completed antiparasitic therapy of 14 days. She soon became unresponsive with a GCS of 5, no gag reflex. There was horizontal nystagmus appreciated. She was again intubated for airway protection, transferred to the ICU. So now our CT, this is our first CT. It shows a significant left to right midline shift of 5 millimeters. There's bilateral white matter edema, and you can see a round mass measuring 3.4 by 2.4 centimeters. And the compression of that left lateral ventricle. So we suspected that this is likely an abscess given the presentation of the mass and its adjacent surrounding edema and mass effect. Again, we consult infectious disease, and antiparasitics are initiated, and we re-pan culture to make sure that there's nothing else going on, there's no new infection. Neurology is again consulted, and the patient is given a loading dose of Keppra and Vimpat for seizure prophylaxis, given their high probability and threshold for seizure, low threshold for seizure at this time. Neurosurgery at this point is consulted, and it's a change in service, so it's not the same individual. So the patient started on mannitol as a Hail Mary, and then 3% saline along with steroids just to kind of decrease that cerebral edema and midline shift. Unfortunately, though, shortly afterwards, the patient, the RN notes that the patient's pupils are unequal. You can see here's our MRI from that same day. There's significant hydrocephalus, there's and ventriculitis. The left lateral ventricle, periventricular area, intraventricular lesions are noted. There's a 3.1 centimeter with surrounding edema, kind of right there in the bottom. And then again, it shows that midline shift. Also, there's right basal ganglia findings that may be indicative of pus. So this does fit in with the abscess. So neurosurgery's interpretation of that previous image was a left-sided basal ganglia thalamic abscess associated with ventriculitis, brain edema, and worsening hydrocephalus. CSF cultures were positive for Streptococcus agalactiae this time, so it was bacterial in origin. Antiparasitic drugs were changed to vancomycin and meropenem. Our CT shows now worsening global edema and mass effect. You can see that there's moderate and hydrocephalus and blurring of the great white matter differentiation at this point. She's neurologically not following any commands. The patient, again, has a Hail Mary, underwent a right-sided burr hole and ventriculostomy with an EVD placed in order to provide some ventricular decompression and relief. This is our follow-up CT after the EVD placement. You can see the ventricles are completely decompressed and flattened. I mean, there's no semblance of them, and there's worsening edema. There's no differentiation of the gyri and sulci here. And then this is the follow-up. These are all serial CTs that were done roughly 12 hours apart. And the midline shift is appreciated. At this point, the patient's pupils remain fixed, dilated. There's no brainstem reflexes, and the patient has a positive apnea test and is declared brain dead. Unfortunately, the patient did not make it in the case, and care was withdrawn. So a brief review. I'm not going to spend too much time on this, but I want to go to the autopsy of our case here. But Tenosyllonium pork tapeworm, typically endemic to Central South America, the Sub-Saharan Africa, India, Asia. Prevalence is typically higher in rural and urban areas of pig farming and suboptimal sanitary conditions, as well as immigrant communities, and that's probably the biggest factor here in the U.S. is along the southern border. That's where you typically will see it. And big note is that you don't necessarily need to have a history of pork consumption or travel. It can be transmissible from fecal oral. I'm not going to harp on this, but again, this is just for just references in any case. But you can see that it can follow two paths of cysticercosis, where you're infecting tissue or the brain, or a teniasis, which you have self-infection of the gut. This is that abbreviated cycle and what I just kind of mentioned. But for time's sake, I'm just going to go on. These are the different life cycles. So neurocysticosis, most common parasitic infection of the central nervous system. Its clinical manifestation really depends. It depends on multiple factors: the stage of infection, whether it's vesicular, colloidal, granular, nodular, or nodular calcified, and then the site of infection, and that can be categorized as intraparenchymal or extraparenchymal, and those can dictate how you treat these. Cyst burden is a major factor, and we'll talk about that in a second. Host immunity in terms of the pathogen's ability to evade the host by disinhibiting the complement system, altering cellular response, and degradation of attacking immunoglobulins, as well as the response to the therapy we're giving, the steroids, the antiparasitic therapy. So intraparenchymal lesions, they're the more common ones, over 60%, and they typically can present 3 to 5 years post-infection. I'm going to backtrack. It's important to know that most people with neurocysticosis may remain asymptomatic, and you may find on incidental CTs multiple calcified lesions of a parasite that has done through its course. So that's why it's pretty common. But these will typically manifest 3 to 5 years post-infection. They can form as non-viable calcified, single small enhancing, or viable parenchymal form, and then there's the rare form of cysticercal encephalitis. The presentation, clinical symptoms you typically will see of this is seizures and headaches, and that has predominantly to do with the increased immune reaction and degradation of the actual cyst. So as that process goes, that's when you have that presentation. You may also have visual and cognitive changes, as well as focal neurologic deficits. And again, symptomatology really depends on the number of cysts and the degree of burden, as well as the inflammatory response. However, it's known that intraparenchymal typically has more favorable prognosis. Extraparenchymal lesions, on the other hand, have very diffuse presentation. They can affect the ventricles, the subarachnoid space, the spine, the eyes, and extraneural areas. Their more scary manifestation is erasmus, which is multi-cyst, neurocysticosis, and these are pretty hard to treat. These are typically associated with a larger parasite burden, and their presentation, their clinical manifestation is increased intracranial hypertension and obstructive hydrocephalus, and this is what is the predominant reason for their generalized worse prognosis. This is our diagnostic criteria. I have the citation here. This is just to complete the presentation. We'll talk about that right now. But on a clinician standpoint, the things that you want to look at that should raise your bar for suspicion is clinical suspicion. First of all, we start with, the patient's presenting with a seizure or elevated ICP. Well, that neurocysticosis needs to be on the differential. You need to have that as at least something that you've thought about. Then the epidemiologic exposure. Have they traveled? Where are they from? What do they eat? And then if anyone else around them is sick, Neuroimaging, specifically to find cystic lesions, brain enhancing lesions, and calcifications is a major plus, and ideally recommend both CT and MRI together. Otherwise, MRI on its own can give you a lot. Serologic molecular testing is a confirmatory test, and then cerebrospinal fluid analysis. Brain biopsy is meant to be a last resort when you're not able to differentiate between the lesion and neurocysticercosis, which is what we ended up having to do in our particular case because we did not get our molecular testing and serology testing in time. And then the ophthalmologic exam can also be pathognomonic. These are the stages that I was mentioning a little earlier, and you can kind of see that the spread and distribution of the cyst can have a different effect. Recall the symptoms are more so noted when the cyst is degenerating and the inflammatory response of the body in response to that. And then this is what you see in a histopathologic side. You would actually see the organism. So had our biopsy shown that, that would have been pathognomonic for it as well. But unfortunately, we didn't have that on our biopsy. So management, you start with antiparasitic therapy. It's albendazole, 15 mg per kg, 2 doses, and this is most efficacious due to its optimal CSF penetration and least effects with medications. Praziquantel is 50 mg per kg divided by 3 in a day, and this can be used in conjuncture depending on the cyst burden and presentation. Anti-inflammatory medications should have a major effect in helping decrease the cerebral edema. Decadron, as mentioned, 0.1 mg per kg. There's also recent studies that show that methotrexate and anti-TNF agents can also be a major assistance in severe cases. However, it's important to rule out tuberculosis or anything like that. In terms of anti-epileptics, the general consensus is carbamazepine and phenobarbital, but it's also noted that this may or may not have much of an effect in seizure prophylaxis. And then our last resort for management is surgical methods like cyst removal and VP shunts, recognizing that that is also potentially an itis for infection as well, but something that needs to be done in an emergent situation. And then to consider differential diagnosis as well, obviously, which, when you work these up, you need to have a broad thought. And as mentioned, most cystic lesions follow a benign asymptomatic pathway and resolve on their own with development of calcifications. However, there are those that just do not, and our body's response to them accelerate the presentation. Treatment has been contentious. There are literatures that say the anti-cystital drugs have been attributed with greater risk than benefit. That's why it's one of those things where you have to anticipate with this cyst burden. With my parasitic therapy and the rupture of cyst, am I going to cause more good than harm in terms of the pushing them towards seizures or obstructive hydrocephalus or so. Mass cystic death leads to extensive inflammatory response and detrimental systemic effects, including increased ICP hydrocephalus. And then neurocranial instrumentation also predisposes to the risk of infection. Steroids can also exacerbate temporary immunocompromised state. And these all kind of go to the autopsy of our case. As mentioned, a brief overview, images preliminary CSF studies were suggestive of infection, but there was no initial organism. The patient was started on broad-spectrum antibiotics because we were trying to rule out septic emboli or bacterial meningitis, but our cultures and echo were negative. The patient did have a pneumonia of strep agalactae, and antibiotics were changed. The patient did complete an appropriate course of antibiotics for that. Burhole was done just to kind of get a biopsy so we knew what we were treating, and eventually that was negative. But serology came back positive for neurocysticercosis. Imaging consistent with intraparenchymal, but the patient was symptomatic and progressively worsening, so they received antiparasitic and anti-epileptic therapy along with steroids. And she showed initial improvement with discharge. However, she came back despite having completed that course of antiparasitics and steroids with a much more markedly worse state. And you saw those CT head images showing the significant cerebral edema and ventriculitis. Our CSF at this point was positive for strep agalactae, and despite measures, she decompensated. So what did we think probably happened in our case? We think that maybe healthcare acquired where she did get that pneumonia, And it's likely that that exposure, along with her extended healthcare stay, which was 25 days alone at our hospital, followed by another 2 weeks at an extended care facility, may have exposed her to that, with the fact that she had the burr hole, she had the ventricular EVD, um, and then her pseudo immunocompromised state with the high dose steroids that was given to her. So it's kind of important to keep that in mind, that nurses' may not be as cut and dry, easy to treat, and it's although there are guidelines, IDSA 2017 guidelines that kind of tell you what to do, but it's a very clinical judgment, and it depends on the presentation of the patient, and you have to have an anticipated approach on what your treatment may do. Um, and that individualistic approach is absolutely necessary in these cases. And despite treatment sequelae such as infection, epileptic states, extended neurologic damage, still maintain a high mortality and morbidity within neurocysticercosis. So, further investigation on maybe post biopsy prophylaxis or EVD or neuromanipulation antibiotic course, maybe that needs to be given into consideration, or patients we anticipate with higher degree of hospital stay, or those with active infections. Um, Regardless, I'm sorry, this may have been a little longer presentation. Do you guys have any questions?
Thank you, Dr. Syed, for your wonderful presentation. Does anyone have any query? Hope there are no queries. Thank you once again, Dr. Syed, for your wonderful presentation.
Mhm.
And by this, we come to the end of day one webinar. I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed the webinar and gained knowledge from the seniors. And I request you to attend tomorrow's sessions too. If you have any queries, you can mail our program manager. Thank you, everyone.